Assalamu alaikum, myself Mohammad Murtazal, and today my topic is DNA, abbreviated as D-ribonucleic acid. The discovery of the DNA was first observed or first came into study in the mid-1860s by a Swiss scientist, Johann Friedrich Michel. He set out to learn more about the components of white blood cells, which are the part of our immune system. Afterwards, Dr. Rosalind Franklin X-ray diffraction at King's enabled the identification of the structure of DNA in 1953. The Nobel Prize for discovering the structure of DNA was afterwards and later awarded to Sir James Watson, Francis Crick, and Maurice Wilkinson in 1962. Although Rosalind Franklin X-ray images of DNA were important in the discovery of the double helix structure and the shape of the double helix DNA. The DNA or the deoxyribonucleic acid is a complicated molecule that includes all of the information needed to construct and maintain a living organism. DNA is found in the cells of all living things. Practically every cell in a multicellular organism has the set of the DNA in which an organism is compiled of, and the hereditary substance is, and the genetic material or the genetic systematic material are being enclosed. As per we further exceed, I would like to illustrate or elaborate the composition of DNA. DNA is basically composed of the monomers nucleotides. Nucleotides are the chemical building units that makes up DNA. It is composed of a phosphate group, a sugar group, and one of four types of nitrogen bases, and also known as the building blocks. Nucleotides are joined into chains with the phosphate and the sugar groups alternating to produce a strand of DNA. The section of DNA or the double helix that provides the structural support to the molecule is known as the phosphate backbone. DNA is made up of two strands that are collected together in a ladder. Well, let's study the structure and the roots of the DNA. Deoxyribonucleic acid is a polymer made up of two polynucleotide chains that coil around together to create a double helix and carry genetic instructions for all known organism and virus formations, functions, growth, and reproduction. Nucleotides are structured in a spiral termed as a double helix, which is made up of two long strands. The base pair operates as the ladder rungs, while the sugar and phosphate molecules serve as a ladder vertical side pieces. Each molecule of DNA is made up of two complementary sequences of nucleotides linked together via hydrogen bonds created by the bases pairs GC, known as a guanine and cytosine, and AT, abbreviated as adenine and thymine. Through use of one DNA strand as a precursor of the synthesis of a complementary strand causes duplication of genetic material. Now, the functions and the operations DNA is capable of, we are gonna study. As we know that DNA is incapable of storing and transmitting genetic information, also referred as encoding information. It governs the cell metabolic activity. DNA is a material that transmits traits from one generation to the next. DNA provides a blueprint for all life's development, evolution, reproduction, and functionality. Protein synthesis is being monitored in the DNA. Now the uses and the capabilities of the DNA are being enlisted. First of all, DNA is being used in the DNA fingerprinting. As per Exceed, I would like to illustrate DNA fingerprinting is a molecular test that reveals a person's or others' living genetic profile. It is used as a court evidence to identify corpse, trace blood relatives, and explore for medical remedies. The second use of DNA or the usefulness aspect of our DNA in our modern society is DNA sequencing. Scientists can use the sequencing to assess what type of genetic information is being conveyed in a certain DNA segment. For evolutionary analysis between species of populations, DNA sequence from various organisms can be compared. DNA sequencing, for instance, can show alterations in a gene that might cause a disease. Polymerase chain reaction or the PCR. It is a technique used to amplify a segment of DNA of interest or produce lots and lots of copies. In other words, PCR enables you to produce millions of copies of a specific DNA sequence from an initially small sample, sometimes even a single copy. Afterwards, recombinant DNA technology. Human insulin, interferon, and human growth hormone are all the manufactured using recombinant DNA technology. Well, these were the aspect and the significance, the components of the DNA and the structure, the elaborated form of a DNA. Thanks for watching.